Could eating less actually slow down aging at a biological level? Caloric restriction isn't just about weight loss. It's a powerful tool that may reshape how our bodies age. For decades, scientists have studied caloric restriction, reducing calories without malnutrition, and research suggests it may slow aging, delay age-related diseases, and possibly even extend life in everything from worms to mammals. But living longer isn't the whole story. That's where healthspan comes in. While lifespan measures how long you live, healthspan is about how many of those years are spent free from disease and decline. Caloric restriction isn't just about adding years to your life, it's about adding life to your years. In this video, we'll break down the mechanisms, the evidence, and what it could mean for your health and longevity.
Caloric restriction and theories of aging. The first of which is the genomic maintenance hypothesis of aging, just that with less calories, there's less growth, and then less required maintenance and cellular damage, namely DNA damage and the accumulation of unwanted proteins, which are natural processes of cell proliferation. Still, the neuroendocrine theory of aging is the most mainly connected to caloric restriction. It involves hormones, both insulin and IGF-1, which is insulin-like growth factor. The idea is that with less calories, there's less nutrient signaling, so there's less insulin and less IGF-1 release, which leads to less growth. An example of this is seen in Ames and Snell dwarf mice, both of which don't have growth factor, and they both have a longer average lifespan and maximum lifespan.
Let's talk about DNA methylation with respect to aging. Now, DNA contains the instructions to make proteins, which are the building blocks of our bodies. And when DNA gets methylated, this can inhibit the expression of the genes. And DNA methylation is highly correlated with aging. So higher levels of DNA methylation means faster aging. Let's look at a study that analyzes the effects of caloric restriction on DNA methylation. Now, this study was conducted on humans with two groups, a control diet and a calorically restricted diet. The researchers used 3 DNA methylation clocks, PhenoAge, GrimAge, and Dunedin pace. And again, these are really accurate ways of predicting age. The researchers observed lower levels of DNA methylation with the Dunedin pace methylation clock in the calorically restricted diet. Looking at the third image here, you can see in red as the calorically restricted diet. Over the course of 2 years, they observed lower levels of DNA methylation compared to the control diet. And this suggests that the calorically restricted diet may be slowing aging. However, the results from the other 2 DNA methylation clocks showed no statistically significant difference between the control diet and the calorically restricted diet. So some of the clear evidence that we see in some of the animal studies may not transfer over to humans.
One study from 2023 tested the drivers behind the lifespan improving properties of caloric restriction. Using C. elegans worms with some genetic changes, the outputs of various enzymes, genes, and transcription factors were tested, all of which are linked to longevity. This helped the researchers determine which were essential in supporting longer lifespans. Tests were then performed to track lifespans among other variables in worms under caloric restriction. In their tests, one main result was that ADH1 extends the lifespans of each test group and proved that is a strong tie to longevity. This prompted the researchers to run further tests using mice and humans in an attempt to replicate the data. They found that ADH1 increased when on caloric restriction in both humans and mice, and that ADH1 overexpression in mice can protect against age-related heart and brain decline. Overall, when humans cut out calories, their bodies produce ADH1, just like in worms and mice. However, some caveats to the study are that they have yet to find causation, meaning that although caloric restriction correlates well with higher ADH1 levels, it is yet to be proven that it drives longevity. In addition, individual variability, such as genetics and diet, is always a big factor that can alter how much ADH1 affects you.
Caloric restriction and type 2 diabetes in humans. So there were around 100 patients in Italy that were put on either a standard diet or calorically restricted diet. Those individuals were either overweight or obese and had type 2 diabetes. For this study, 25% caloric restriction was selected. Compared to a standard diet, after 6 months, those that were on a calorically restricted diet had significantly lower BMI, waist circumference, triglyceride levels, and GFR, which is glomerular filtration rate, which is how well the kidneys filter. And in this case, a lower level is desired for those with diabetes. The study was actually done for 24 months, and BMI, waist circumference, and triglyceride levels were still significant between standard diet and the calorically restricted diet, but for GFR, wasn't significant like it was for 6 months, but still there was a lower level compared to baseline. And additionally, only the calorically restricted diet individuals showed a significant increase in HDL compared to baseline. That's good cholesterol because it takes cholesterol from the organs and then brings it to the liver to be metabolized.
This study looked at caloric restriction in genetically diverse female mice. There were 5 groups, including a control, a 1-day per week fasting group, a 2-day per week fasting group, and then a 20 and 40% caloric restricted groups. They found that the 40% caloric restricted group had the greatest extension in lifespan, but that genetic background was actually more important in determining lifespan. There was a higher incidence of greater loss of lean mass, along with harmful impacts to the immune system. And so this shows us that there's a difference between lifespan and health span, and how a intervention can potentially impact both of those things. One caveat is that this was only performed in female mice, so it may have neglected any sex differences in the results that could have been seen in male mice. From this data, our group concludes that caloric restriction can be effective for extending mean and maximum lifespan. However, there needs to be further studies done to analyze how caloric restriction affects health span. There is some conflicting evidence which displays that it might not have as beneficial impacts on health as it does longevity. Overall, there seems to be a proven benefit to longevity and lifespan, but the effect on health span is currently too unpredictable. We recommend that there be further studies to determine the specific impacts that caloric restriction has on health span in humans.